morning everyone. We will present the Microscopy 2 titled Slide Preparation in Microscopy. This activity focuses on learning how to prepare microscope slides and how to properly observe different plant specimens. Our main goal was to identify the basic parts of plant cells, such as the cell wall, cytoplasms, nucleus, and chloroplast, and compare how these structures look different plants. We examined 4 specimens such as onion, digman or hydrilla, tomato epidermis, and squash stem. This activity gave us hands-on experience in slide preparation, staining, and microscope focusing, which are essential skills in studying biology. For materials and methods, we use the compound light microscope, dissecting kit, glass slides, cover slips, dropper, iodine solution, and the plant samples. For the procedure, we follow the steps such as set up the microscope properly, ensuring that the light, mirror, and objective lenses were aligned. Prepared such specimen by taking very thin sections. For the onion epidermis was peeled from a fresh onion layer, and digman or hydrilla was taken from a single leaf. For tomato epidermis was carefully sliced from the outer skin, and a thin cross-section of squash stem was taken using a blade. Each specimen was placed in the glass slide, stained with iodine to improve clarity, and covered with cover slip. Then we observe all samples under low power objective or the LPO and high power objective or the HPO to examine their cell structures and detail.
Onion specimen. Under the low power objective, the onion cell appeared as a multiple triangular cell arranged in the net and brick-like pattern. They looked transparent and thin, but the cell walls were clearly visible. Under the high power objective, the structure became detailed. The cell walls were more defined, the cytoplasm became clearer, and depending on the stain, the nucleus sometimes become visible as a darker spot. Onion cells are a classic example of plant tissue because their clean and rectangular shape that naturally show the cell wall very clearly. The nucleus may not always be visible unless the dye reaches the nucleus may not always be visible unless the dye reaches the right area. This is normal for onion epidermis.
The second specimen is digman, also known as hydrilla or elodea. Under the low power objective, the cells appeared green and aligned in rows. The cell walls were visible and the overall structure looked like stacked bricks. Under the high power objective, one of the most interesting things we observed was the movement of chloroplast. The green dust chloroplast could be seen slowly circulating around the inside of the cell. This movement is called cytoplasmic streaming, a process that helps the cell distribute nutrients. Hydrilla is a perfect specimen for studying live plant cells because the chloroplasts are active. The movement we observe is proof that the cells are alive. This specimen is very helpful in visualizing chloroplast and cytoplasmic flows.
Third specimen is the tomato epidermis. Under low power, we saw many cells with noticeable and thick cell walls. Unlike onion cells, the tomato epidermal cells were irregular or polygonal in shape, not rectangular. Under high power objective, the cell wall became very distinct. Tomato epidermal cells are very different from onion cells because of their irregular shape and the presence of pigment. The thicker wall also makes them easier to see even without heavy staining. The last specimen is the squash stem. Under low power, we could see the general shape of the cell, though the details were not very sharp yet. Under high power, the cell became much clearer. We also noticed green pigments within the cell and trichomes. There are tiny hair-like structures located on the plant surface. Their trichomes have important function in plants. They help protect the plant from insects, reduce water loss, and sometimes reflect too much sunlight. However, in our observation, the nucleus inside the trichomes was not clearly visible because the staining was not strong enough.
For question and answer, 1. How do cells of the onion specimen appear in the microscope? Under the microscope, the onion cells appear as elongated, rectangular cells arranged in a neat brick-like pattern. Their cell walls are clearly visible, showing the typical plant structure. The nucleus may appear faint or sometimes not visible depending on the staining and focus. Number 2. Are the cell walls thick and the nuclei visible in the onion epidermal cells? The cell walls are distinct but not particularly thick. They are clearly defined and separate each cell from its neighbor. The nuclei are not always visible in every cell. They may appear faint or absent under low power mode. But under high power, some cells might show a small dense nucleus if the staining is adequate. What shape do the epidermal cells of tomatoes exhibit? What is the color of the dominant pigments here? The epidermal cell of the tomato are irregularly shaped polygonal, unlike the uniform onion cells. The dominant pigment color observed is yellowish or light orange due to the presence of carotenoids in the tomato's epidermal layer. Number 4. Did you notice in the movement of chloroplast? Describe what you have observed. Yes, in the Hydrea specimen, you can observe cytoplasmic streaming where the chloroplast appear to move slowly around the edge of the cell. This circulation helps distribute materials within the cell and is characteristic feature living plant cell under. 5. What is the shape of Hydrilla's apical and marginal cells? The Hydrial cells are elongated and rectangular in shape. Arranged in the long rows, the apical cells at the tip of the leaf are usually smaller and more rounded, while the marginal cells along the edge are elongated. Both contain numerous green chloroplasts, giving the sample its bright green color. 6. In plants' trichomes, did you see prominent nucleus? Is the trichome septate? In the pictures we took, the prominent nucleus was not visible in the trichomes. It was also unclear whether the trichomes was septate or non-septate because the details were not clearly shown under the microscope. In conclusion, the microscopy activity allowed us to clearly observe, compare, and identify the major structure of plant cell, from onion, digman, hydrilla, elodea, tomato, and squash stem specimen. Through proper slide preparation and the use of both low-power LPO and high-power HPO objective, we were able to distinguish key plant cell parts such as cell wall, cytoplasm, nucleus, cell membrane, and chloroplast. The onion cells showed organized brick-like arrangement. The hydrilla displayed active chloroplast movement. The tomato epidermis revealed a regular polygonal cell with visible pigments, and the squash stem provide a view of various tissues including trichomes. Although some details such as nuclei in certain samples were difficult to observe due to staining or focus limitations. The activity successfully strengthened our microscope skills, deepened our understanding of plant cells and structure of diversity. Overall, the laboratory exercise enhanced our observational ability, improved our competence in slide preparation, and provided valuable hands-on experience in studying plant tissue under the microscope.